So I just wanted to do a really quick overview of stem cell and tissue engineering as a whole. So maybe I was going to ask the audience to raise their hand. That can be electronic or visual. If you've ever heard of stem cells, or if you haven't, so raise your hand if you have heard of stem cells before. Yeah. Okay. Cool. So stem cells have been growing in popularity, and they basically can be turned into any kind of cell in the body, namely three types: ectoderm, endoderm, mesoderm. And this just means that stem cells can create a plethora of cells in your body. We used to think that stem cells could be only found in embryonic fetuses, and then recently, within the last 10 to 20 years, we found out we can actually induce pluripotency, which are now called iPSCs, or induced pluripotent stem cells. And so what's really neat about iPSCs is that you can differentiate them down any kind of lineage. That could be cardiac cells, lung cells, skin cells, brain cells, you name it, we can do it. And so our lab uses iPS cells to differentiate into cardiac cells that we can mature and basically differentiate in the lab that will emulate what cardiac cells do during development, which is beat really strongly. And we use this to basically make regenerative patches for cardiac injury. And this can be congenital heart defects that are born when you're, or that are found when you're born. This can also be cardiac patches during myocardial infarction or heart stroke. And so what's really cool about this is what Duncan was talking about is he's using hydrogels to study different types of disease pathology. But you can also use hydrogels to, as an injectable substance to kind of help a heart, like a stroke after a heart, or a heart after a stroke. Sorry about that. So that's basically what our lab does, is looking into regenerative medicine using stem cell therapies to help the correction of congenital heart defects. But other labs can also use it to restore heart function. And so my role specifically as a bioengineer and student researcher is all around cardiac defects, found specifically in Down syndrome. And so this was an animation here that I made that's basically just showing you that the heart has these four cardiac regions, two atria and two ventricles. And the purpose of these atria and ventricles and their separation is so that you can have separate flow. And so blood flow that goes to the right side of the heart is actually delivered to the lungs, at which point it is oxygenated and then returned back to the heart, where it can be systemically pumped throughout your body. And so this is great. This keeps oxygenated and deoxygenated blood on separate sides. However, in the case of some disease pathologies like Down syndrome, we see that half of kids born with Down syndrome are also born with congenital heart defects. And this can be a heart defect between the ventricles here on the bottom, a heart defect between the atria here at the top, or a combination of the two. And so what happens here is that you have blood from different sides of the heart crossing over, such as this, which basically decreases your stroke volume and increases your heart rate. So you have to constantly be working so much harder to pump your blood throughout your body. And so the question that my research looks into is, how do these septal defects form in Down syndrome? And to look at this, you kind of have to go back to development. And so we see that the heart is the first organ in the body to develop, and it begins as these two little heart tubes that fuse together and then do a series of curling to ultimately define these four regions that we know to be the atria and the ventricles here on the bottom. And so for those cells to separate and basically form a wall between the top atria and the bottom ventricles, the cells from the center and the top have to migrate down and upwards to fully form that septum to allow the separation of deoxygenated and oxygenated blood. And so specifically in Down syndrome, a really smart group of people decided to do a chromosome mapping study using cases of partial trisomy 21, where only part of the chromosome 21 is triplicated. And they found that of those kids who had Down syndrome or partial Down syndrome and had a heart defect, a huge candidate gene on this chromosome is called discam, or cell adhesion molecule gene, and collagen 6. And so collagen 6 basically upregulates the adhesion processes during cardiac development, so it makes it really hard for those cells that are migrating and dividing to actually do so, because they're kind of stuck in this glue. And so to study this, what I personally do in my lab is I use iPSCs to model cardiac septation in Down syndrome. And so we are able to get, this is kind of strange, but the samples that we get are urine samples. And so when you pee, you're actually shedding urine epithelial cells from your renal system. And so those can be collected and then reprogrammed using induced pluripotency factors that ultimately allow us in the lab to use stem cells. And then so my work specifically is using these stem cells from Down syndrome patients and control patients to differentiate into cardiomyocytes. So they're these beating, functional, kind of immature cells, but they're beautiful to look at. And so what I use them for is looking for differences in proliferation and migration, specifically on collagen 6 and other kinds of extracellular matrix proteins that will prevent the migration and proliferation during cardiac septation. And so what this looks like in the lab under the microscope is actually quite phenomenal. You can see these cells spontaneously beat without any kind of physical interruption at all. I'm not touching them. They're doing this all on their own. And sometimes it just looks like that, and sometimes they get a little bit crazier. Or even crazier. So these discs are amazing, and they're absolutely so much fun to look at in the lab. What I can do with these cardiomyocytes that derive from stem cells is I can stain them using different kinds of antibodies that are specific for cardiac cells. And so anything that you see as bright orange is a cardiomyocyte, and anything you see as blue is a nucleus or basically the DNA is held. And so I have a bunch of different iPSCs from a bunch of different Down syndrome and control individuals, and I can differentiate them all into cardiomyocytes. So control 1, control 4, these are from different human beings. And so what I do then is I basically lay these cells on different extracellular matrix components like collagens and fibronectins and things like that. And then I can take pictures of them over time as they migrate, and then do something called tracking. It's called a TrackMate plugin on ImageJ, but basically it just creates these vectors on the program that allow me to track where the cell has gone, how fast it's gone, and how far it's gone. And this other video isn't showing up, but it's basically just the same thing, watching these cells kind of migrate over these substances. And so using this technology, what we're able to do is quantify the velocity and displacement of these cells on different substrates. So namely, this is a control, this is just kind of the basic seeding components that we use in lab, but we also use type collagen 1, fibronectin, and then the really interesting one I was interested in for Down syndrome is collagen 6, because it is upregulated in trisomy 21 and has been shown to predispose kids to heart defects. And so what you see here in black is that the iPSC cardiomyocytes from Down syndrome individuals migrate significantly less far, so they displace less, and they also have a lower mean velocity, indicating that upregulation of collagen 6 in heart development can actually hinder the migration of those cells, which can predispose you to a heart defect. Additionally, we can look at how the cells divide. So if you see these green little dots right here, those cells have divided. The blue dots are also DNA, and the green dots are also DNA, but the green ones have just divided or proliferated. And so what I can do is seed these cells on different substrates, like collagen 1, fibronectin, which are commonly found in the heart during development, and also collagen 6. And so you see here that the cells on collagen 6 from Down syndrome individuals actually proliferated a lot higher than other substrates. And so this is a part of my ongoing research that's pretty exciting. I actually have some experiments going on as we speak, so it's all culminating into something really exciting. But what I also wanted to talk to you guys about, since this is bioengineering is always evolving, always changing. This was a video that really got me excited when I was entering the field. So yeah, you heard him right. He's taking vegetables. And what their lab does is they use different techniques of decellularization, using different kinds of soaps and things like that to remove genetic material from biological substances. And so you can use spinach leaves, you can use pigs, anything. You can decellularize it and take out the DNA from the matrix. And then what you're left with is this really interesting scaffold. And in the case of spinach, we have a big problem in bioengineering of creating vasculature in our bioengineered scaffolds. And so what the spinach leaves do is they already have vasculature which allows blood flow to flow through the scaffold. So this is something I thought was crazy. They're just taking everyday materials and making it into something kind of Frankenstein-y. So that I thought was really cool. And we actually replicated this in our own lab for a high school summer camp that we did last year. And the students thought it was amazing because they could see the material fading from the leaf over the week. So really cool. And then something else that's relevant to what Duncan was talking about is 3D printing. And so this group in Israel, basically this group in Israel was able to bioprint using biological materials to print a heart. And so they're able to create different chambers and things like that to ultimately create it's more pretty than anything else, but it's basically just to show you that the bottom line is research in bioengineering. Because that research is incredible, especially in bioengineering. The field is constantly evolving. And being a researcher as a student allows me to kind of ask my own questions, find my own answers, and be surrounded by incredible people while I do it. So with that, I would like to thank everyone who came, and I'm open for questions as well as everyone else. But yeah, I just wanted to thank you guys. This has been really fun.